On the left, I will navigate to Molecular Biology Tools, to Golden Gate. In my basic setup, you can see my scenario is set to entropy plasmid set. However, there are other options such as combinatorial assembly, advanced assembly, and split sequence. For assembly restrictase, my restrictase I've chosen is B-cell. However, there are other enzymes you can select. Click Next. Now I will upload my backbone. I will select PGGA, and I will also upload my parts. Click Assemble. The image shows a circular plasmid map of a genetic construct named PGGA with fragments 1 through 6. The map features color-coded segments representing different genetic elements with entry plasmid set parts newly introduced on the right side of the diagram. As I scroll to the right, you can see my sequence as well as the assembly parts that were introduced and the enzymes. We can register this new plasmid. You can give it a name and review the sequence here. Click OK. I will run an advanced search. I will filter by date created and run my search. Here you can see the new DNA part that we've just registered. You can see my plasmid to the left as well as the sequence to the right, where you can view the assembly parts as well as the various enzymes. Here you can see my annotations, the label, start position, end position, and the annotation type. Streamlined Golden Gate assembly with versatile entry plasmid sets in electronic notebook like this one orchestrates modular DNA construction and expedites synthetic biology innovation.